Hi, this is Dr. Jabali, and in this video we're going to talk about proteins, which is included in the topic 2.4. Here are the understandings, applications, and skills that we need to know for proteins. We definitely understand how amino acids eventually become polypeptides, which are basically large molecules of protein. We're going to look at all the different amino acids there is. We're going to see how they're actually linked together, their sequence. We also going to talk about genes and how genes can influence the sequence of the amino acids. We're going to talk about the shape that the amino acids can take, and that their shape eventually tells you their function. So there's the 4 level. Although this might be like an HL content, I do like the SL students to be strong about it because it kind of gives you a leverage when it comes to answering questions. And we do talk about other types of proteins such as globular and fibers and right at their functions and structures and so forth, as well as many other proteins. Nevertheless, we're also going to talk about proteome, which basically is it's all different types of proteins that come out of a single gene, for instance. And of course, never forget, we have a certain proteins that we must know according to IB. We have to know their functions. Also, we're going to know how pH and heat, these two, they can affect the shape of the protein. Or otherwise, if they go out of the optimum, then they're actually going to cause a denature. And nevertheless, we need to know how to draw an amino acid with its peptide bonds. So this is what the video is about. So let's go forward. So first we have to know proteins. What are the elements that creates those proteins? Every single protein will have for sure these 4, CHON, carbon, hydrogen, oxygen, and nitrogen. Those 4 are there. There might be other things, like a phosphate, a sulfur and stuff, but these 4 are the most common elements in all the amino acids. There is about 20 essential amino acids. You don't have to know all their names, but you should know that there's 20 amino acids out there. And when I say amino acid, we are talking about the monomer. That means the 1 unit of a protein is an amino acid, and this is 1 unit right here. Now, I'm going to draw eventually in the next slides how to draw an amino acid, but this is one of the structures that you must know for IB is the amino acid. Now, they do have a very primary function, and it's the construction of the body structures. That means everywhere in your body that has structures, for example, like your muscles, these have proteins. Now, right here is a very general representation of what a protein looks like. You can see that these little squares right here, these are actually the amino acids. And what's in between them, it's right here, these bonds. And these bonds are actually peptide bond. Now, peptide bond is only between amino acids. You will not have peptide bonds on glucose or lipids or nucleic acids. Peptide bond is just for proteins. So, moving on, since now we have to really know how to draw the amino acid because this is something that is very common to come to IB. Here is the picture of an amino acid. Now that's a very generalized. Now we have to learn first this each side. Right here on this corner, usually on the left side, we have the amino group, and you have to know this name. And in this side, right side, we have the carboxyl group. So it's always amino group on one side, carboxyl group on the side. It doesn't change. It's always that. Now, looking at the amino group, you notice that there is a nitrogen and 2 hydrogens, while carboxyl group, a carbon is bound by a single bond to oxygen and oxygen to a hydrogen. Sometimes they do draw like this. So it's totally fine too. And then you have on the other side a double bond with another oxygen. Now in the middle, you have a carbon connected to the 2 sides, to the amino group and the carboxyl group. And you have a hydrogen going up, and then you have the R. Now, what is the R? R is the variant. The variant basically means it can be switched. It's like a substitute. Like in a game, a soccer game, you want to switch a soccer player for another player. You just switch, and then that means you have somebody else with a different name coming in. Now, since we kind of got a general idea, now we have to be able to draw it because that's the most important thing. So, when it comes to drawing, it always starts on the left side. You put a hydrogen attached to a nitrogen and to the hydrogen. This has the amino group. Then, of course, there is the base, which is the carbon here. The carbon in the middle will be bound to a hydrogen and to a R group. And then there will be another carbon, which is from the carboxyl group, which will be bound to OH, or you can extend the H if you want, and then an oxygen. Some places they flip up the oxygen on the other side. It doesn't matter. As long as you have shown the carboxyl amino group, you're completely fine. Okay? Now, Here are the 20 amino acids. You can see this entire large structure here. Now, where exactly will this larger structure include? This larger structure would actually been right here on the R. That means this R here, you will take this R and you will replace this entire structure. And that will give you arginine. Or let's say you want to take this structure right here, it would have been cysteine. Or if you take this structure here, it would have been valine. So what you do is you just replace the R with one of these structures and you get the 20 amino acids. Now, the good news is that you do not have to memorize any of these structures. That would have been a killer, but if you go to college for biochemistry, you should know them very well. But you do need to know that there is essentially 20 of them, and they are replaced with the R group. Now, we have to know how to build a protein, right, and break a protein. Now, right here, we have 2 amino acids. Now, if we want to use the word condensation, which you already learned this in topic 2.1, condensation is when you're building it up. When you're building it up, what's coming out of it is water, okay? But when it comes water, where's that water coming from? That water is coming from, again, one of the hydrogens here, okay? And it's taking the OH here, the hydroxyl, we would say, these two together, and together they will form the water right here. When they do this, then where's the bond going to be? Now, since the bond is gone here, it's the carbon that is going to bond to the hydrogen. So it would have looked like something like this. You have hydrogen with the nitrogen, just like the one in above, a carbon to a hydrogen in the R group, and then you have this carbon, which is this. Now, since these are all gone, we have a double bond oxygen. I'm going to put up here. Now, it needs to have a bond with the nitrogen here. And then we have a hydrogen that is left, right? This hydrogen was left, this was gone. And then you connect to the hydrogen, the R, and then the carbon with the O and the OH. So you can see here that I have connected the 2 amino acids. And this right here, always between a carbon, right, and between a nitrogen, this is where you find the peptide bond. So there might be questions where they put this long chain of amino acids and they say, spot, where do you find the peptide bond? And the peptide bond will always be between a carbon and a nitrogen. So always spot for that carbon and nitrogen, and that's where you find the peptide bond. Now, this arrow right here, it just means that it's going in the direction of condensation, right? Now, if we want to use the other arrow, which is the arrow going up, that means the water here is going to be coming into this area here and breaking that peptide bond, and it will go and look like this all over again. So what would happen? Oops, let me go back again. So we have the hydrogen here, and we have the carbon, and we have the double OH. So what happens is if the water binds here, that water basically means there's an OH and there's an H. So one of the H will go for the nitrogen right here, and the OH will go to the carbon, and they go back to being this. And the hydrogen that came back to the nitrogen will be that. So water actually splits those two. When it splits, that means the arrow is going up, and this will be known as hydrolysis. So hydrolysis is when you're breaking down a large chain, and condensation is when you're making a big chain. That's 2.1. I'm just reviewing it, but I'm showing you with an amino acid group. Now, let's learn a little bit more about the structures and the functions of the protein. Now, whenever we're going to say, what does that protein do? The first thing we do is look at its structure, because the structure is the one who's going to tell you what are you going to be, what are you going to do. Now, that structure also depends on the sequence of the amino acids. So here you can see there is different amino acids, Ser, Val, Ser, Glu. That's a chain of amino acids, keep going, right? That's the long chain. Now, down here, look at this. We have different types of species, gorilla, chicken, right, iguana, bullfrog, and shark. Now, you can look at the sequence of their amino acids, and you can see that they eventually have differences. Like all of them have ALA, if you can see here, but then we have tyrosine right here, which shark doesn't have. That means the structure of the shark is already changing from the rest. Then they all have LEU, right, right here, and they all have GLU. But then you can see GLI here is different And then the three here have met, and there's little. So if I keep going, you will notice that none of them has exactly the same chain of amino acids, right? There's somewhere that they are different. Like you can see, I can highlight the differences here. This one is different here. This is different. These are different. And then let's see here, difference here, difference here. And then we have a difference here, difference here. So you can see that these differences shows different sequence, and if different sequence, then eventually they're going to have what? Different structure. That's why a gorilla does not look like a chicken and a chicken does not look like iguana, because they don't have the same sequence of amino acids. They might have some, but for them to be exactly the same, that means they should be 100% exactly the same sequence of amino acids. Now, besides looking at the sequence of amino acids, that's usually it is the primary structure of a protein, we're going to look at the secondary, tertiary, and quaternary, because there's 4 levels of organizations that protein structures can go. What I just did here was only focus on the primary in the sequence of these amino acids. So let's look at these 4 levels. I just mentioned about the first one, which was the primary level, right? As you can see, here is just a straight line, right? But eventually that straight line starts to fold. When it starts to fold, it turns into beta sheets and alpha helix. Now, the beta sheet and alpha helix, they're folding because there is an important thing that is found there in order for them to fold, and that is the hydrogen bond. Oop. And then once you go to the tertiary level, you might have alphas, like you can see, right? You have the beta right here and you have the alpha. Here, you have a mixture of these two. But here, besides them having these alpha and beta sheets, you can also have other types of bonds besides the hydrogen bond. And we can see that this can have a disulfide bond, right? This can have ionic bond, can have another covalent bond, and so many other bonds. And that's why you can see that it's more folded in this way. When it comes to quaternary structure, it's not just one chain. It's not this chain only one. It can be, this is a chain, 2, 3, 4. You can see there's 4 chains right here. And in these 4 chains, you can have alphas and betas and different types of bonds. So this is a very quick looking at it. And here you can see it. You're going from a primary to a secondary to a tertiary. It's like every time you're building up, you're getting more things, like you're getting more bonds. For example, here on the first one is just a peptide bond. And then here you add the hydrogen bond. And then here you might have so many others like ionic, covalent, disulfide bond. And then here is where you have more than one chain. That means you might have 2 chains, 3 chains, 4 chains of amino acids. This is how it's built up. So let's look closely on each one of them. So the primary level, as you can see, is just a chain of amino acids. And you can see that they are bound by one thing, peptide bond. So the sequence of the amino acid tells you which structures and functions you're going to get, okay? And sometimes, here's the bad news. If you, by mistake, put the wrong amino acid in there, the entire structure and function goes away. And that's what happens with sickle cell anemia. So sickle cell anemia is just one change in amino acid, and our red blood cells does not function anymore. Imagine, it could be thousands and thousands of amino acids, and if you just switch one, that's it. The red blood cell doesn't work anymore. That's example. So that's primary. Then we get to the secondary. The secondary is where it's not a straight chain, right? Now the chain is doing like a little curve, or it's doing a zigzag, right? What are these curve zigzags? Is because there is bonds happening, and let me try to highlight here so you can see what I'm talking about. You see there's a bond happening here? These are hydrogen bonds that it's happening, hydrogen bonds. So what is this hydrogen bond being? It's basically it's a hydrogen bond connecting one amino acid to another. So it's not just peptides, but it's actually, and it's usually happening between the nitrogen and the oxygen of double bond. Do you need to go details? No, you just need to know there's an additional hydrogen bond that allow this little turn like this, right? And the same thing here. You can see hydrogen bonds, hydrogen bonds, hydrogen bonds, and that allowed it to fold like this. So the folding in this case is a beta sheet, And if it's a spiral, it's an alpha helix. These are two examples of secondary level. Now, if you notice, I did not mention anything about the R groups because they're not playing with the R groups. It's usually between a nitrogen of an amino acid connecting to the oxygen of the other one. You see? So it's a bond that is happening here between them. That's usually where connection is. That means the hydrogen of that nitrogen bonding to the oxygen. Let me make this more clear. So you got the nitrogen bond to a hydrogen, right? And on the other side, you have the carbon bond to the oxygen. So it's this one right here. It's the double dots between those two. So for the HR students, you're already getting the hang of this thing. And why did I put a double dot? Because again, this is a hydrogen bond that we always never do a hydrogen bond. We do dots. We do not make a straight line. So anyways, when it comes to secondary level, we have to remember there's alpha and beta sheet. These are two examples of secondary. That means one is spiral and one is a zigzag. So here you can see it's a hydrogen bonds. As I was talking, you can see hydrogen bonds, remember, between the oxygen and the hydrogen of the nitrogen. These are hydrogen bonds that are connecting between different amino acids. And you can see there's also here. So that's the secondary level. Then we get to the tertiary level. When it comes to tertiary level, not only do we have the alpha and the beta, right? That means the hydrogen bonds and the peptide bonds. We are now interacting with the R groups of the amino acid, right? And you can see in this picture, like we have so many different types. If you look at this, you have a hydrogen bond here, right? That could have been the spiral, right? And then here you have a hydrophobic interaction. That's another type of bond. And then you have a disulfide, which is two sulfurs. So you say disulfide means two sulfurs bond. And then we have ionic bond, which is a charge of this right here is an R group of one amino acid, and this is the R group of another amino acid, and they're connecting with each other. So it's an R of an amino acid connecting to another R. That's the connections that gets to the tertiary level. Now, these connections can be covalent bond, such as the disulfide bond. We got van der Waals interactions, ionic bonds. So we just don't have the peptide bond and the hydrogen bond. Now there's an addition of covalent bonds, ionic bond. And this time we actually are used. The most important thing you need to know is that the R groups right here, these are the ones being connecting in the tertiary level. Now an example of a protein that is found in the tertiary structure is enzymes. So all the enzymes that you're going to learn in IB here, they all have the tertiary level, which that means they have hydrogen bond, peptide bond, disulfide bond, ionic bond, all covalents, van der Waals, all these bonds, and they're connecting the R groups. So looking more closely, let's see here again, right? We have these three bonds. It's disulfide bridge right here, ionic bond. We can also have the hydrophobic interactions, the hydrogen bonds. So you can see not only is there was a bond between like the peptide bonds here, right? And the hydrogen bonds, but there's additional bonds. And at the same time, you can see that there is bending going on, which that means they might be alpha or they might be beta. So tertiary has both. And then we get to the last level, which is the quaternary. Quaternary means the fourth level. So here, we're not just made of one, it's not one chain of amino acids. You actually have one and a two and a three, sometimes four, and then they come together. So it can be more than one chain of amino acids. That's why it's called multiple polypeptide chains. Like this one example, you can see there is four colors. That means there are four chains of amino acids connecting. And inside of those, you have alphas, you have beta. It's like you're building up from the primary and the secondary and the tertiary. So you're taking their hydrogen bonds, you're taking their peptide bonds, you're taking the disulfide bonds, you're taking so many things together. In addition to all of these guys, it has a prosthetic group, which basically, these are called conjugated proteins. What it means by prosthetic groups. We're talking about iron. Okay, so iron is basically a mineral, right? And it's a metal. And the metal is right here. It's not part of the chain. It's just an addition to it. It could be calcium. It could be vitamins. So when I say prosthetic groups, we're not talking about amino acids. We're talking about additional things to the amino acids, such as iron, magnesium, calcium, vitamins. These are additional to it. Now, hemoglobin, this is for IB. You do need to know that hemoglobin is an example of a quaternary organization. Why? Because this picture right here, it actually shows you hemoglobin. Hemoglobin has four chains, and it has the iron right here, and this iron right here is the one that attaches the oxygen, because again, Hemoglobin is found in the blood. And what does the blood do? The blood carry oxygen. Now, so how does the blood carry oxygen? Well, because inside the blood, there is the hemoglobin, and hemoglobin has iron, and iron gets it. So hemoglobin, what does it have? It has iron. And what does iron do? Carry oxygen. And since hemoglobin is in the blood, voila, you have the quaternary organization. So just to be clear, the only examples that we have to know for these levels is that enzymes are at the third level, okay? And the hemoglobin here is the fourth level. These are the two examples that you must know for IB. So wrapping up again these four levels, you see the first level right here is just a straight chain. And when you start bending them, they become a secondary, which is the alpha or the beta, right? The beta sheets. And then after this, if you start using the R groups to interact, they become a little bit more folded, right? And then eventually, if you add more chains, they become the fourth level. So these are the four levels that we must really know for IB. Now, let's talk about polar and non-polar because this is very important. Sometimes you get to a chapter. Now, we already learned from water, right, that polar means that they have a charge, a plus and a minus charge, right? And non-polar, that means they're neutral. That means there's no charge, there's zero. So some amino acids, they might have charges and some amino acids might not have any charge. And then some amino acids depends on the chain. Like you might have a chain here, right? A chain of amino acid. And right about here is positive, and maybe here's negative, and then here's a positive, and then a negative, negative. Whatever it is, then throughout the chain of the amino acid, it has charges. Now, just as a recap, when I say hydrophobic, it means it does not like water. And if I say hydrophilic, it means it loves water because the word hydro means water. And philic means love, and phobic means fear. So sometimes in this long chain of amino acid, like for example, some areas here, okay, let me erase this because this is not true. There might be like amino acids here, right? And if these bunch of circles are amino acids, right? So right up from here to here, there might be zero charge. And then from here to here, might have positives, negatives, and positive. So this part would have been the polar side of the amino chain. And this part would be the non-polar. Now, why would you want to have a chain of amino acid which sides have non-polar and polar? Because here is what you do. When you fold this chain, right? Let's say I use this chain. I'm going to do a circle. When you fold it, voila, like this, and then you fold it like that. What does this look like? This actually looks like a channel. Like a channel to allow, whatever molecules you want to do to go in, right? The molecules go through this channel and get inside the cell, right? But if you remember from the chapter on membranes that you have the phospholipids here. Now, very important that you know here. Look at this. This is a fatty acid. It's made of fat. This is fat. Okay, so if you have a protein channel, because this is a protein, right? Channels are made of protein. How can that protein be touching that green part? How? It's impossible for that part which is completely polar be touching something that is non-polar, right? Because fat is non-polar. So what's the truth is, on this side of the protein here, it will be known as non-polar. And this side of the protein will actually be polar because you'll be touching the water, right? And the whatever materials that are coming in. So if you see this picture right here, which can come in the IB exam, right here what we see is a channel. And you can see that the channel is entirely made of protein. But what you see here, the pink part of that protein is actually non-polar, which that means it's hydrophobic. It does not like water. And that's good because that means it can touch the fatty acids of the phospholipid. However, you see the green part right here of the protein channel. It's actually touching the fluid outside of the cell, the fluids inside of the cell, as well as the molecules that are coming in polar, right? So they have charges. So that will be known as polar because they have the plus and minus, and that means they are attracting with the water, which has plus and minus. So it's very important to remember that protein channels, pumps, all these guys, on a certain section of the protein, it will be non-polar, hydrophobic, and there will be a section that is hydrophilic, and that's where it allows the molecules to go in. Now, there are some of the proteins that you must know their functions, basically, because I be asked you. Now, throughout the year, for example, there's some of these that you will see if you're doing HL students. Unfortunately, from some SLs, you will have to know what they are, very generalized here. So, for example, hemoglobin, which you just learned, it's actually a protein and it carries iron, and that means what it's doing? It's transporting oxygen. So, again, hemoglobin is fighting the blood and it's carrying that oxygen. Now, actin-myosin, that's an HL content, but these are actually proteins and they're found in your muscle. So they do muscle movement. So actin and myosin are muscle movement. Insulin, you probably heard this in life with diabetic people, right? Insulin is actually a hormone, but it's also protein. So it's a hormone made of protein. And what it does, it maintains the blood sugar, right? It's coming from the pancreas. You will learn this in the digestive system. What it does is it releases insulin, and insulin makes sure that the sugar is in a good level, because too much or too less can cause different types of diabetes, right? So again, insulin is a protein hormone. Then you have immunoglobulins, which is, this is for immune system for the HL students, but you need to know here that it's also protein, and basically it's fighting bacteria and viruses. So immunoglobulin is an antibody, right? It's an antibody that is fighting bacteria and viruses. So it's found in the immune system, which is a defense system in our body. And last and not least, the amylase is an enzyme, and all the enzymes are proteins. And what does it break down? It breaks down starch, which you will learn in the digestive system. So in this topic 2.4, you still haven't got to the digestive system, which is topic 6. But once you get there, you're kind of going to know these guys really well, most of you guys. But the main thing is just know their functions. Very simple. So again, hemoglobin has to do with transporting oxygen in the blood. Actin-myosin has to do with the muscle movement. Insulin is a protein hormone, deals with the blood sugar. Immunoglobulin has to do with attacking bacteria and viruses. And amylase is an enzyme that breaks down starch. If you can put that as a summary, you already got yourself the points. So we can talk about protein acting as different things, right? Because protein can have different functions. So proteins could be a structure. If it's a structure, it could be a collagen or a spider silk. If it's a hormone, it could be insulin and glucagon. If the protein is working in the immunity, it can be hemoglobins. If the protein is a transport, it can be a hemoglobin, right? He's transporting oxygen. If it's sensation, it's rhodopsin. Rhodopsin is what you find in your eyes and you're sensing the things around you, light, for instance. Movement, it can be actin-myosin, which is the movement of the muscle. And then enzymes, we have Rubisco, which is in photosynthesis, and catalase, which can be broken down in our own body. So these are the different types of functions that protein can inherit. So a protein can be just for structure, a protein can be a hormone, a protein can be working in the immune system, a protein can be transporting things, a protein can sense things, a protein can move you, or the protein can be an enzyme that is basically digesting. So there's different ways in which protein can actually function, and here are examples for each. You need to know this. Now, there's two types of proteins that you must know about their shape and about their solubility. That's the fibrous and the globular proteins. So the fibrous ones, as you can see here, it looks a lot more like this, and it's very narrow shape, and it's not soluble to water. So for example, collagen and actin, these types of proteins, they do not dissolve in water, okay? They stay intact. On the globular proteins, they're more like a 3D round globular shape, right? Because it's the word globular, you can see it's a globe, it's a circle. And it's water soluble, that means it does dissolve in water, it moves through water. In this case, we have hemoglobin and insulin. So this is the maximum you need to know. And you can see that the shape right here is more like a globular, while this is more like a narrow. So these are the only things you need to know between fibers and globular proteins. Their shape, are they soluble, not soluble to water, and then give an example. If you know this, you got yourself already a good enough information for fibers and globular proteins. So make sure you do know examples, okay? Do not just know if their shape is a narrow or globular, if they're soluble to water or not. Also figure out examples, because sometimes IB likes you to know these examples, okay? Now let's talk about genes and proteins and how genes can influence proteins. Now, here's the most important thing, and eventually in the future we'll know, is that genes do not control you, okay? What controls the genes is the environment that you're living around. It is the thoughts that you have. So for instance, here we have one gene, let's say you have that one gene in your body. And then you are exposed to vitamin A, to environment A. In this environment, it let's say it has a lot of heat. If this environment has a lot of heat, then that gene will produce protein A. Now, let's say the same gene is exposed to an environment that is B, and that environment has a lot of, let's say, wind. And if that wind hits that gene, the exactly same gene, it's not going to produce A. That environment is going to produce a protein B. If you are in an environment C, and that environment C, let's say it has a lot of trees, so that gene is being exposed by a lot of trees, then it's going to produce a protein C. So it's not that the idea that one gene can only produce one protein. That is not true. Okay, it's not one gene, one protein. That is not true. That one gene right here, that one gene, can produce millions of different types of proteins. Why? Because it's exposed to millions of triggers. So depending on their stimuli, that's what's going to tell the gene what is going to come out of it. Okay? So this is where we get to the word genome and proteome. Right here, we have the word genome, and genome basically means all the composition of the genes of that human body, right? And proteome is all the entire sets of proteins expressed by that genome, by that organism, by that tissue. Like, what are all the proteins that can come out of that one gene, right? So here we have in our body about 20 to 25,000 genes. And of course, they will have different types of stimuli, right? Different types of environment pushing on us. And depending on the environment, you will produce a certain type of amino acid chain, and then eventually it will turn to different types of proteins. So you can see, you can have just because of that one gene, you can have so many different options of protein. And that's just one gene. So imagine you have 25,000 genes, and each of those 25,000 genes, you got a chance of producing 1 million different proteins. So you can actually make the calculation. And that's why we all this, let's say, 6, 7, 8 billion people, why are we're different, nobody's almost the same or exactly the same, is because, again, we are expressing our genes differently, even though we have the same gene, right? The same gene. Again, even though we have the same gene, the way we express is different because each of us are not experiencing, not living the life exactly the same, thinking the same thing, doing the same thing, acting the same thing, eating the same thing, talking the same thing. We're not doing that. Everybody's unique. That's why our genes are expressed unique and we don't look the same. So if there was two people, okay, we all have the same genes, by the way. Remember, there's 25 genes. We all have those. All of us have those exact 25 genes, 1,000 genes. However, the way we express is different. So if we have two people who are going through the same life, the same thoughts, the same thinking, everything exactly the same, then they're going to be a clone. But you never have two people who have exactly the same thing in every second of their lives. That's why they'll be different. So with that in mind, remember, proteome is just all the sets of proteins created by that genome, which is all your genes. We talk about proteins, and now let's talk about denature. What does that word denature mean? De here means off, nature. That means you're getting out of your nature. In this case, we're talking about the structure of the proteins. So that means the protein is losing its structure because of some manipulation. Like for here, you can see a protein has been folded into a structure that will give him a function, right? He has his structure, he has his function. If it goes through denature, you can see it gets unfolded. So it lost its structure. If it loses its structure, so again, if it loses its structure, it loses the function. So it won't work anymore. That's what denature means. It just destroys its function. Now there's two ways in which you can denature any protein. In this case is the temperature and the pH. Some enzymes, like for example, let's use us humans, right? We have our temperature of 36 degrees. That's our body temperature. So that means our enzymes are working around that 35, 37, maximum 38, right? That's how our enzymes are working. However, if our body temperature rises from a 36 and keeps rising to a 38, 39, our enzymes are starting to be destroyed. If we get to 41, we're having a fever, we're going to the hospital. If we have 44, if our body temperature is 44, my friend, we're done, we're finished, we're killed. Why? Because that temperature, 44, kills all our enzymes, because our enzymes has this optimum. And when I say optimum, that means its best moment of work, which has to be at 36. So if the enzyme is not working at a temperature of 36, he's done, he's dead. We're basically doing what? We're destroying the hydrogen bonds. If you remember that from the secondary structure, we are destroying the bonds of the proteins. So temperature, you got to be very careful. Some enzymes like to work at the temperature of 30. Some enzymes look at the temperature of 36. Some work at 50. Depends on the animal or the plant, whatever it is. They have a specific optimum. When I say optimum, in their best temperature, then they work. But if they move out of that temperature, they're most likely evil to nature. Same thing goes with pH. Now, pH, we have the acids and the bases, right? And we know that the amino acids has positive charges on them. Now, here's what happens. If you change the pH, right, away from what they want, you are changing the charge. So let's say proteins, a chain of protein was like positive, 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 positive. He's always positive. If he goes to, in his pH, the pH that he likes to work is at 2. But if you shift the pH to 3, what is he doing? Is destroying these guys. It turns into negative, turns to neutral. If that happens, that's it. The enzyme, or in this case, the protein, will not work. Because the protein will say, hey, dude, I can only work at pH of 2. If you put me at a pH of 4 or 1, I'm done. I'm denatured. My charges are messed up and I cannot function. That's why even the pH is very important to proteins. Proteins are sensitive to pHs and temperatures. They have to be exactly at the level that they want, otherwise they will denature. And this is pretty much the topic 2.4 when it comes to proteins. Here are some of the questions that you can go back and read them and go through the video again because they have been answered for you. Okay? And it has everything you need to know about them. And that's pretty much. Thank you.